Well, thanks everybody for coming today. Welcome to the California Academy of Sciences and welcome to Science in Action. My name is Jeff and I have the distinct pleasure of joining all of you today as well as one of our researchers here at the Academy, Gary Williams. Can we all give Gary a big round of applause for being here today? Thank you. Very special treat that we get to do about twice a month, which is being joined by our researchers at this program called Chat with the Academy of Scientists, second and fourth Wednesday of the month. So Gary is the curator in our research department here called Invertebrate, Zoology, and Geology, which is kind of a mouthful. And I imagine a lot of you know what that means, but just to kind of clarify for maybe those who don't, what is invertebrate zoology exactly? What does that translate to? What's that? No bones. So animals that don't have any backbones. And that's basically the group under which Gary falls, though he studies kind of a more specific group than just animals with no backbones. And that is, Gary, specifically corals, and even more specific than that, octocorals. Is that correct?
Correct.
All right. So how is it that you first got involved with studying kind of such an esoteric group of animals as octocorals? And what led you to decide to devote your entire career to studying those animals?
Well, I think it started with, I was fortunate enough to have a great high school biology teacher, just a few miles north of here, north of Brooklyn in Fairfax, San Antonio, California. And he was the guy who stressed morphology of different animal groups. He was a marine biologist who got us out in the field, and that instilled in me a real interest in all sorts of different animal groups in the marine realm. We did field trips to tide pools, and that was one of the things that started it off. And then later I was able to study the biodiversity of coral fauna of southern Africa, the South African Museum in Cape Town, and that research I used for my PhD thesis later, so that's how that began.
All right. So since then, you've devoted to your career to studying animals that tend to live in shallow water areas. So you've also studied animals in much deeper water areas.
Yeah, it's actually deep water and shallow water. When I was doing in South Africa, we did a lot of deep water research too, using dredges and trawling off of research vessels. So not just coral reefs.
Right. So here we have a picture of a coral reef area, which, of course, is in the shallow water area, and right next to the rainforest. So this is kind of a nice little comparison here, because one of the things that we often say in our coral reef dive programs is that in some ways the coral reefs are almost like the rainforest of the sea in how much biodiversity they're able to maintain and support. So what is it really about coral reefs in your own words that you think allows them to support such diverse life?
Yeah, diversity, there's a lot of theoretical answers for diversity, and a lot of the answers are complicated. One of the reasons is because corals are symbiotic organisms. They have algae living in their tissue, therefore they can use sunlight to develop nutrients, food resources. That gives them enough energy to produce lots of calcium carbonate, laying down a huge geological structure to be used in a lot of different organisms, different microorganisms. Our coral reef is multi-layered because it's such a huge structure, and a lot of organisms can inhabit that structure. There's a couple of reasons. Other reasons have to do with isolation of coral islands, and isolation is one of the ingredients you need for evolutionary change. Diversification. So these are some of the ideas that we have for diversity in coral reefs.
So this next slide here actually shows a couple images that were taken out in the field on some of your research expeditions. You do go out and actually do very hands-on work as part of your research. You actually go out and dive in the field and study the areas where the animals live that you're studying. So that's a pretty cool perk. That's a pretty awesome reason to become a invertebrate zoology curator, I think.
Nothing to complain about.
Right. So what is it, when you're actually going down here and doing these things, not only looking at the creatures, but also taking a couple samples to bring back to the lab with you, what is it that you're actually doing? What's a typical day like out there?
This is underwater research where the Academy has huge collections of organisms behind the scenes here, and we use those for biodiversity research, and we get loan requests from around the world, and scientists come to use our collection. So one of the things we do is to collect the organisms. That's the first step. Next step is to study them to see if you have new species. How does it fit into phylogenetics or evolutionary relationships? How does it fit into biogeography and overall evolution? So we start by collecting the group that we're working on. And here we're using, we take underwater photographs of everything, plus the fact that we take a small sample of the coral where we don't harm the coral. The corals that I work on are usually branched animals that climb up high, and we just snip off a piece that we need. Never take the entire organism. So, and we bring them back to the lab in alcohol and put them in a collection for future study.
And in your time down there underwater, you'll encounter a whole variety of animals. Of course, there's tons of different things going on, not only corals, but things like sea anemones, starfish, sponges, lesser-known things like sea fans and sea pens. So you get to know a lot of these animals pretty intimately. Are there any specific things about some of these animals that you find really fascinating and really makes you really appreciate what it is that you're doing?
I think overall it's the diversity. There's so much variety in our life form and color, and feeding and reproduction. Everything is different. There's always something new for a curious person with a keen observation eye. It's just a matter of diversity and variety that I think is the most important thing for somebody who's interested in it.
Have you ever encountered something that was pretty surprising? Maybe something that you hadn't expected, maybe an adaptation that was pretty new to you?
We have found bioluminescence and things. Bioluminescence is where an organism will give off light, produce its own light, and give it off for a defensive mechanism. And we found that in several corals at night in caves. We had no idea we're able to do that. And that was by accident. One time I went into a cave and brushed up against the side, and the whole cave lit up green light. And I said, I have to find out why it's deciding after green light, and I selected the organism that did. So that was a discovery of something that was totally unexpected.
Did you find out why?
Did you find out why
it's lit up?
Why defensive mechanism. What we think bioluminescence in corals has to do with defense. Like if a fish tries to take a piece of the coral and it flash and shocking the fish.
Do all corals have the bioluminescence?
No, only a few octocorals as far as I know.
So here's a couple other images of different animals that you're studying here. I think people could be excused for thinking that corals are plants, because they certainly look like them an awful lot. This is a coral up here, coral down here, and a sea fan, I believe, up top there. Yeah, so they're really amazing animals in that way. Can you describe actually what is the animal here that we're looking at? This is a whole colony of animals, correct?
Yeah, this is a soft coral. And each one of these pink areas is a bunch of polyps. Those are the individuals, the feeding individuals. So it's an animal because it feeds on other organisms. And you can't see the polyps here. You can see these are polyps. Tentacles, and the tentacles have stinging structures in them that kill microscopic animals. That polyp will bring it to the mouth and they digest. So in the old days, 200 years ago, they thought these things were plants until somebody observed them actually eating other animals. They figured out they're actually animals.
And here's some of the other exotic animals that you've encountered. This one here over here looks like an urchin, but this is actually a type of sea star. Is that right?
Yes, that's kind of a thorn starfish, and this is a destructive agent of coral reefs. These starfish actually eat the corals that build coral reefs. Sometimes the populations will explode and coral reefs can be decimated. That's happened in the past, but it's actually localized, and as the population goes down over time, the coral reefs come back. So it's not permanent damage, it's usually temporary.
And the function of the thorns, is that a defensive thing?
Yeah, those are typical of venomous substance, and we've been hit by those, and if they penetrate your skin, you can get a big reaction. Some people are allergic to it, so yes, it's a defensive thing.
And what is this animal over here?
That's a sea pen. That's a coral that lives in soft sediment at the sea bottom. Sea pens are distributed around the world in deep water, abyssal plains, and shallow water too. But because they can adapt to soft sediments, they have a huge distribution and not restricted to coral reefs or rocky areas. They go down to like 6,000 meters, which is almost 20,000 feet in the ocean, where it's black.
Is there a question over here?
So those are each individual polyps on
yes. Each one of those is a polyp.
Sea star. What are the predators? What each starfish starfish. We don't have big ones.
What eats the sea pen?
No, the starfish.
That's probably nothing as far as...
Yeah, no predators. When you have that many poison tipped thorns, then that is
They're the predators.
Yeah. So this is like a fish, a worm that eats them.
There are some starfish are eaten by some snails and parasitize them. Most of them are tough enough that they have a defensive mechanism, so they are eaten for the most part. Fish will try and eat small ones.
So, of course, the work really only begins once you're back from the dive. You have so much more work to do from that point. And here's some pictures from out in the field, post-dive. Some of your colleagues here doing some of their initial lab work. What is it actually that has to get done the moment you return from a dive?
The main thing is looking at live animals. So you bring them up and you make your original field notes under the microscope of live animals. Because you can't see the polyps and corals when they're dead or preserved. You see them when they're alive. The thing you do is as soon as you come up and do your field research in the field lab, it's makeshift, whatever you can find. You bring a microscope in your bag on an airplane. Sometimes it works, sometimes it doesn't. And then you set up a lab and have a look before you go back.
And that's the office. That's pretty nice.
That's not the office.
Okay. Okay. Very good. Well, one of the things I think is really fascinating about your work is that you've been doing it so long and going to some of the same locations for over a period of time. You've been able to see things change over time. So I'm wondering, what are your impressions about how some of the environments that you've been to over the years, how you've seen them change?
I think as far as change, the reefs basically stay the same. What changes is the amount of bleaching that we call coral bleaching. And that's what we've noticed. Before 1995, you hardly ever saw anything as far as bleaching. Once in a while, maybe. After 95, almost every dive trip, or every dive, you can see some example of bleaching. What bleaching is where the algae that live in the tissues, which are golden green color, they leave the animal, the coral, because of the high temperatures. The sea temperatures are too high. So they leave, and once they do that, the skeleton of the coral is white, and that's all that's left, and it looks like it's been bleached. It doesn't mean the coral dies, because when the sea temperatures go back to normal, they can be reinfected with the algae, and they're back to normal. If the sea temperatures stay high for a long period, the coral can die. And we're not sure what's going on. El Niños would do that because of the high temperature. As far as global warming, the jury's still out on that. We're not really sure how that affects. But it happens all the time now. It happens more since 1995 than it did before 95, as far as we can tell.
And, of course, the Philippines has recently become a major center of not only your work, but of researchers from many different departments here at the Academy. And actually in May, there's an expedition coming up, one of the largest expeditions that the Academy has ever gone out on into the Philippines. It's called the Hearst Expedition. And so researchers from a variety of fields will be going out with you and doing deep water research, shallow water research, and research on the land and terrestrial research. Can you tell us a little bit about what the main goals of this expedition are and what particularly you're going to be doing out there?
Basically a biodiversity survey. It's what part of the world that has probably one of the highest diversity areas in the world. It's part of the Coral Triangle. The Coral Triangle is made up of the Philippines, parts of Indonesia, and New Guinea. And the Great Barrier Reef, which everybody's heard about, has about 2/3 of the diversity outside that triangle. The Philippines, because there's so many islands, there's over 7,000 islands there, that has an incredible diversity. And you go from one island to another, and it changes. So that's why we go to the Philippines. We've been going there for a long time. The coral reef tank you see down here is based on our research location. That's what we get. The whole thing is based on what it looks like when we go out there.
And what is it that are your particular goals for this upcoming trip?
I'll be involved in the two of those, the shallow water and the deep water expedition. Shallow water is to do coral reef research for biodiversity surveys. That's exactly what we've been talking about. The deep water component is on a research vessel where we'll be taking trawls and dredges to get deep water samples to do the same thing, biodiversity in the deep sea, which is even less known in the coral reefs.
So when you and your colleagues go out there, are there any ways in which you kind of work with the local community or work with the local governments to help conserve some of these areas? What other type of conservation work do you get engaged with?
Yeah, first of all, we have to have the permits to do all this